me a little bit more, and then I'm done talking. So, Dr. Elia Shahbazi received his bachelor's in applied mathematics and master's in pure mathematics from Sharif University in Iran, and then went on to co-found 2 startups and worked for around 10 years. He then sold his companies and moved to the US and received a second master's in software engineering from North Carolina, followed by a PhD in computer science and neuroscience from the University of Notre Dame. His thesis was about computer-aided tools for neuroscience and neural volumes. He's currently a postdoc in Dr. Arash Afraz's lab, where he's focused on understanding how the brain works through brain manipulation. Fun fact about Elia is that he's a professional photographer, podcaster, startup CEO, dancer, and calligrapher, and his work in brain manipulation aligns with his family name, which translates to king manipulator. We're happy to have him here at Fans today. Please welcome Elia Shahbazi.
So, today I want to talk about the seeing through the brain. But before that, let's look at this. Who is this guy? As Maya said, I received my bachelor degree in applied mathematics, then a master degree in pure mathematics, and then another master in software engineering, and then a PhD in computer science and neuroscience. And as you can see, there's a gap between the 1997, when some of the people in this room wasn't actually in this world, and this 2018 when I received my PhD. But all of these years, I just try to just use all of these years' knowledge to just find out exactly how does the brain work. And there is a question how that brains work, and we can just find it out by brain structure or just knowing about the brain function. So either way, if we want to just do increase our knowledge about the brain structure or brain knowledge, I think we will just find out this, the images and the brain. The difference between these two, I think, is just 2 small prepositions. Images of the brain and images in the brain. So I try to just answer that question with both images of the brain and images in the brain. The first part is the images of the brain. I started my PhD with a project, the IAPA project called Microns. It's a machine intelligence from the critical networks. The goal of this project was to find the structure of the brain by analyzing the macroscopic images of the brain and make the brain blueprint. Let's say there is a circuit in the brain, and all this synaptical connection are the circuits. So we want to find out that how they are just connected to each other and make a structure for that. So these are the things we're using for that. These are the X-ray images between the thalamus and cortex, or the X-ray images in the rat brain. Other one is 2-photon macroscopic images. So with these images, we want to just reconstruct the whole things. We have the myelinated axons, we have the blood vessels, we have the cells. There are different things. So what's the obstacle to just make that blueprint just really fast? Is even for really small images, for example, a 300 by 300 by 100 stack of images, this is really small. If you want to just use a deep learning or machine learning to just segment all of one of those macrostructures, let's say just cells, you need to just train that deep learning for 145 minutes for a really small stack, or 98 minutes with a unit. So it's imagine that if you want to just do it for the whole brain, it will take years. I made a pipeline called Flurry that can do same thing in 25 seconds. So it's way faster. So imagine that if we want to just do a good X-ray, macrocity X-ray of the whole brain in the mouse, it will be 4,000 by 3,000 by 6,000 something pixels, the whole thing. So it's 10,000 times bigger than that really small area that we did with the unit or unit 3D. But fluorescent segment and 2D reconstructed in just 1,000 minutes. And not only just one structure, but the 4 of them, the cells, vessels, myelin axons, and the dura at once. So it's fast. You must wonder just what's the Flurry? Flurry is flexible learning-free segmentation and reconstruction of the neural volumes. If I wanted just to use just the first letters, it wasn't just make sense. So I call this fluorine because it's easier to say fluorine. But the fluorine is faster than the deep learning in order of magnitudes. It's more accurate than machine learning, and it segment and analyze different modalities. So you can just use the electron microscopic, X-ray, two-photon microscopic, light sheet microscopic, expanding microscopic, and everything, and different microstructure in them at once. So it's good. Let's look at some of the 3D reconstruction with fluorine. This is the butterfly brain and visual system. So this area is the brain. These two are the visual system. I used to call them eye, but they're not exactly eyes. And all of those dots are the cells, and all the purple area is optic chiasma in the visual system. I did this with the University of Chicago, and I was in the University of Notre Dame. This is the whole rat brain with the X-ray reconstruction. You can see the blood vessels and the myelinated axon underline, and the durometer, something the rat does. And this is a part of this that you can see just the blood vessels there and the cells, the somal cells there. This is the one that took the 1,000 minutes to just reconstruct the whole 4 things. And it's not just a reconstruction, but also fluorine can report the number of cells, the minimum distance or average of distance between the cells and blood vessels, or the cells and average distance between blood vessels and the myelinated axons. These kind of reports can come from fluorine. And this is the good one, the octopus visual system with the X-ray. It's a joint work with Harvard, and the octopuses are awesome. They're really cool. They have a distributed brain in their legs, so working with them is fun. So, but that also, just doing this in the macroscopic level, you can use fluorine to just make a reconstruction of something like an MRI scan. This is coming from the MRI scan in the NIH. As you can see, we constructed a skull, brain, arteries, and even that vitamin E that you're just putting there. And the whole reconstruction and making this really fancy thing took about 1 minute or 1 minute and a half. So it's really fast. You'll drag and drop the MRI, you will have this in no time. Also, we use the same thing for the brain lesion. J1 gave me an MRI image. It's just 50 slides, so it's really low resolution, but we use it to just find the area that have the brain lesion. And you can see, you can just cut that area, you can just go back and forth in this 3D. Also, the other things we can report is, for example, there is this lesion area is 20% of the whole brain, something like this. So as you can see, you can just see how you can cut this and find the different areas in that. So fluorine is fun, but it took a lot of time to just make that thing, and people trying to just do their imaging better. So I couldn't help them in that. So while my proposed method is just being used different places, they are using it in Oregon National Lab, University of Notre Dame, Harvard, and Chicago to use this fluorine to just make this cool 3D things. But they moved to NIH to just do the better things and the things that actually work. So my work is, what we want to do is finding out how these images are in the brain. So from the images of the brain, we want to just move to images in the brain. And we know that with just targeted, for example, the targeted electrical stimulation, we can just change things in the brain. For example, there is a really cool work in Stanford that Dr. Parvizi just used this targeted electrical stimulation in a 47-year-old patient. And he found out that this electrical stimulation will just change the faces, so they are just mess with this facial perception. Let's look at this 1 minute, 2 minute movies. I don't know if you have. Okay, let me just check. 1, 2, 3. So this is for the sham.
I'm going to do it one more time. Look at my face. 1, 2, 3.
You just turned into somebody else.
Tell me
Your
about
face metamorphosed. Your nose got saggy, went to the left. You almost looked like somebody I'd seen before, but somebody different. That was a trick.
Could you see my eyes in place?
I could see your eyes, but you could have been somebody else who had, well, he has similar eyes to Dr. Parvizi, but you were someone else. Your whole face just sort of metamorphosed.
Did it get warped?
Yes.
Did it keep its own features?
No, it's almost like the shape of your face, your features drooped. Yeah. Sort of like they drooped to a particular direction, kind of stretched and drooped.
All right. Ready? 1, 2, 3.
This is the less. It's clear.
Yeah, it metamorphosed again, and you look like someone I've seen before, but maybe a different person in my memory. Almost like your nose kind of shifted to the left a little bit, and your look just changed.
Okay. But the problem is, none of these people coming to us and said, okay, just put these things in my girlfriend's or boyfriend's brain. I want to just look really cool for him. When he's looking at me, I want to look like James Dean or someone. So we need to just work with monkeys. So, but what's the problem with working with monkeys? This is a monkey looking at the screen, and we are trying to find out what's in his brain. The problem with the monkeys is they're not talking to us. For some reason, they don't want to talk to us. And we don't know what's going on in their brain. So what we are doing here is we are showing some images to them. And yes. So we are showing some images. And we're just showing these images